Good morning everyone, we are Group 2. Today we are going to talk about genetic engineering. Movies often show genetic engineering technology used by crazy scientists to fulfill their evil plans. Therefore, people may have negative impression on genetic engineering. But what actually is genetic engineering?
Genetic engineering, also called gene modification and genetic manipulation, we use biotechnology to manipulate the gene directly. The manipulation of gene aims to change the DNA sequences of organisms in order to correct some natural defects. This technology is widely used in different fields like medicine and research. There are three major methods in doing genetic engineering. The most common one is the plasmid method. This method is using small circular pieces of a DNA molecule to operate, which is called plasmid. The second method is using the technique similar with the plasmid method, which is called vector method. This method uses vectors, which are the small carrier molecules, which are normally viruses. The third method is called ballistic method, which is also called the gene gun method, which is using gene gun to modify the genes.
One of the applications of genetic engineering is to produce insulin for the patients suffering from diabetes. Another application is genetically modified food. This enhances the quality of the food produced. For example, the GM tomatoes have longer shelf life than normal ones. Let's move on to the CRISPR.
CRISPR technology is the short form of clustered regularly interspaced short palindromic repeats. It is a family of DNA sequence found in the genomes of prokaryotic organisms such as bacteria and archaea. CRISPR technology is a simple yet powerful tool for editing genomes. It allows researchers to easily alter DNA sequences and modify gene function. Its many potential applications include correcting genetic defects, treating and preventing the spread of disease, and improving crops. RNA harboring the spacer sequence helps Cas proteins recognize and cut foreign pathogenic DNA, and other RNA-guided Cas proteins cut foreign RNA. CRISPR are found in approximately 50% of the sequenced bacterial genomes, and nearly 90% of the sequenced archaea. And these systems have created CRISPR gene editing that commonly utilize the Cas9 gene. As is a family of DNA sequences found in the genomes of prokaryotic organisms such as bacteria and archaea, these sequences are derived from DNA fragments of bacteriophages that had previously infected the prokaryote. They are used to detect and destroy DNA from similar bacteriophages during subsequent infections. Hence, these sequences play a key role in the antiviral defense systems of prokaryotes. The CRISPR-Cas system is a prokaryotic immune system that confers resistance to foreign genetic elements, such as those present within plasmids and phages, and provide a form of acquired immunity. This editing process has a wide variety of applications, including basic biological research, development of biotechnology products, and treatment of diseases.
Here are some applications of CRISPR, including gene silencing, embryonic stem cell and transgenic animals, and epigenetic editing. The technology must overcome challenges before reaching clinics. There may be off-target effects, and it must be compatible with the immune system. So CRISPR can edit DNA, but what if I want to edit RNA? Are there any related technologies? Actually, there is. Let's move on to Libra.
Next, we will be talking about Libra, leveraging endogenous ADAR for programmable editing of RNA. Libra is a genetic engineering technique for editing RNA. Mechanism includes engineered strands of RNA called arRNA recruit native ADAR1 and ADAR2 enzymes to swap out different compounds in RNA. These enzymes change a specific adenosine into an inosine on a transcribed RNA, resulting in a change in protein synthesized during translation. Differences between CRISPR and Libra are as follows. Target molecule in CRISPR is DNA and in Libra is RNA. The number of components in CRISPR is 2, Cas9 and single-strand guide RNA. In Libra is only one, arRNA. The proteins that CRISPR relies on are exogenous proteins, and for Libra, it's native proteins. The Libra does not directly affect DNA, so it is not heritable, hence it is relatively more ethical than CRISPR. The number of components affects the delivery. Libra has only one component, making it easier to deliver. Lastly, CRISPR relies on exogenous proteins, which are rather risky as they can result in deviance from cell activity, delivery limits, and immunogenicity, making Libra, despite relative new technology, more safe.